This is Kaushika Patel, an assistant professor from the Department of Pharmaceutical Technology. I welcome you to the MOOC series, an initiative started by LJ Institute of Pharmacy, Ahmedabad. In this session, we are going to discuss about microscopy. What is microscopy? It is a science of observing the organisms by using a microscope. Discovery of microscope is one of the important discovery in the field of microbiology. So let's begin. Microscope means to see is an device which helps to view the small organisms which cannot be seen by the naked eye. And microscopy is the science of investigating such small objects by using microscope. The two important parameters required in microscope are magnification and resolution. Magnification is the enlargement of the original image, that is increase in the length, breadth and diameter of an object. Magnification is obtained by objective lens and ocular lens, and the total magnification is the multiplication of the magnifications of both the objective and ocular lens. What is resolution? Resolution is the ability to isolate the objects in the view. That means the ability to reveal the closely adjacent structure as the separate entities. More the resolution, better is the view of the object in the microscope. Limit of resolution is the minimum distance that is required between two bodies to observe them as the separate bodies. So there are two types of microscope based on the source of illumination. Light microscope that utilizes the sunlight or the artificial light for illumination, and the second is the electron microscope that utilizes the beams of electron for illumination. Light microscope, they are further of four types: bright field, dark field, phase contrast and fluorescence. Whereas electron microscope, they are of two types: scanning electron microscope, that is SEM, and transmission electron microscope, that is TEM. So let's see all these microscopes one by one. So what is bright field microscope? As the name suggests, here the background is bright and you can observe a dark image in that bright background. There can be two types of objective lenses. There can be simple microscope, there can be compound microscope. In simple microscope, there is only simple magnifying lens. In compound microscope, different series of lenses can be used. So this is the image of compound microscope. What is the principle? Here from the light source, the light passes through the condenser lenses and it is focused on the sample that is lying on the platform. The light is scattered by the sample, it enters into the objective lens, then in the ocular lens, and finally the image can be viewed. You can see a dark image in the bright background by using this compound microscope. What are the advantages? Any living or stained cells can be observed easily. It is a very simple setup and very cheap equipment. But what are the disadvantages? You cannot observe the sample without staining due to low contrast. Sometimes by staining, the original morphology of the object or specimen may be destroyed. The limit of resolution is very limited, that is 200 nanometer. Second is dark field microscope. This is opposite. Here the background will be dark and you will be able to observe a highly bright image in the dark background. The biggest advantage is you don't need to stain the object. Without any staining, you can observe the specimen in the dark background. It will use a high intensity lamp and a dark condenser to obtain the image. So you can see that the light source passes through a dark field stop, then it passes through the condenser lens. A hollow cone of light is produced that goes through the sample into the objective lens, into the eyepiece, and then the image can be viewed. Such microscopes, they are used for specific purposes like to observe the motility of leptospira, or to observe the spores within the bacteria, or to observe Treponema pallidum. It is a simple setup. It provides contrast directly to the unstained tissue, so living cells can be observed and their movements can also be observed. But the disadvantage is the strong illumination lamp which is used, which may damage the delicate samples. So this type of image is observed in the dark background. Third is the phase contrast microscope. Here, as the name suggests, you have to generate the contrast between the specimen and the background. Such microscope is used to study a transparent specimen so that the living cells and their phenomena can be studied in the natural state. Here, the light rays in the phase will produce a brighter image, and the light rays out the phase will produce a darker image. It contains an annular diaphragm and a phase plate as shown in the diagram. This annular diaphragm that allows only a hollow cone of light to pass through the condenser and falls on the specimen, whereas the phase plate is used between the objective lens and the ocular lens, and it generates the phase difference by reducing the wavelength of the light. So you can see a bright image and comparatively darker background. Such microscope is used to visualize the internal components to identify and diagnose specific type of cells in the specimen. You can observe growth, the dynamics and metabolic processes of the cell, and any type of thin specimen can be studied. But what are the disadvantages? You cannot study thick specimen by using phase contrast microscope. And the annular ring which is used, it may reduce the resolution capacity of the microscope. So such image is observed in case of phase contrast microscope. Fourth is fluorescence microscope. In this microscope, the specimen is stained with fluorescent dyes. Such dyes, they get excited upon exposure to UV light, and when they come back to the normal state, they emit the visible light, so that a fluorescent image of the specimen can be obtained in a dark background. Here, the fluorescent specimen is kept. That fluorescent is excited by using the UV light. It passes through the emission filter, and then a fluorescent image can be observed of the specimen so that it can be studied easily. Next is electron microscope. Here, the biggest advantage is it uses a beam of electron which have a very short wavelength compared to the light. So, compared to light microscope, the magnification power is very high. Light microscope gives you magnification of 2000 times, whereas electron microscope can give up to 2 million times magnification, and the resolution power is also very high. So first is scanning electron microscope. SEM gives you a three-dimensional view of the surface of the object, which is black and white. So you get a black and white image of the surface of the specimen. The specimen is scanned, is plated with the gold, and that gold plated specimen is scanned. You can observe the surface morphology very clearly by using this scanning electron microscope. Its resolution is 1000 times better than the light microscope. So here you can see that through the electron gun, a beam of electrons is focused, is accelerated by using an anode. It passes through two different magnetic lenses and falls on the sample. This primary electrons, they generate a secondary electrons which are detected by a detector. That detector converts the electrons into an image, and that image will be observed on the screen. So a signal is generated that is transmitted on the screen. This image, such type of image will be observed. This is Bacillus megatherium, and you can see that its surface morphology can be seen very easily in the SEM image. Next is transmission electron microscope, TEM. Here stream of electrons is used which passes through the internal part of the specimen. That's why you get a 2D image of the internal structure of the specimen very clearly. Its resolution is even higher than the scanning electron microscope. So here you can see that a beam of electrons will be generated. They will be focused on the sample by using a condenser lens. Then the image passing through the sample, it passes through the objective or magnetic lens, and that image is transferred to the fluorescence screen by using a projector lens. So finally you get to see a two-dimensional image of the internal part of the specimen very clearly. One thing that is taken to be care is the entire lenses and the sample, they are placed in a column with vacuum and air is completely removed, because if air molecules are present, they collide with the electrons and a clear image is not generated. So such type of TAM image is observed where the internal portions can be studied very easily. So you can say that SAM gives you 3D image of the surface morphology, whereas TAM gives you 2D image of the internal portion of the specimen. The magnification and resolution of TAM is very higher compared to scanning electron microscope. These are the frequently asked questions from this microscopy topic. I hope you have liked the session. Thank you for watching the video. For more such updates, like, share and subscribe our YouTube channel Pharma Ignite and press the bell icon for such more notifications. Thank you and see you soon in the next lecture.